So thank you for the invitation and the nice introduction. What I want to do today is to go a few years back where we got interested in the mechanism of theta rhythm during REM sleep in the hippocampus, and then summarize what we did in those early studies and how this has evolved to more cortical mechanism of REM sleep, or at least brain function associated with REM sleep. So you will hear about a little bit of oscillation, but mainly single cell dynamics in head restraint sleeping animal. So I put few slides. I guess some of you are probably familiar with some background concept on sleep, but I put few slides just to make sure that we're talking about the same thing. It doesn't work. No. So it doesn't work here either. Good. Yeah. So one first statement I will make is that all animals sleep. There have been some debate recently whether some more simple organism like Drosophila or C. elegans, or you might have seen those videos on octopus, actually do sleep. I strongly believe that they do some sleep-like states. Whether it's the same as the one in human, I'm not sure, but I'm happy to discuss that further. But let's consider that all animals sleep. And if all animals sleep, one question that also is important is why do we sleep? And that's the slide that basically summarizes first why we study sleep, right? So one aspect is to understand the basic mechanism of sleep. So this is still something that is not clear. We don't know how the brain and the body communicate to each other to control transition from wake to non-REM and then to REM sleep. Another aspect that is quite important is the brain function that is associated with sleep, or sleep function, if you will. We'll talk about this today, and I will focus mainly on synaptic plasticity, but there's many other functions. We'll have a slide on this, but it's still not clear. Again, we know that some brain function seems to be associated, but what is the mechanism and why they are associated is not always clear. And so we believe that a better understanding of both the basic mechanism and the function of sleep is really important to design new diagnostic or therapeutics for sleep disorder. I listed here some of those sleep and circadian disorder. But interestingly, and more, there is a amazing clinical study that showed that subtle change of sleep and sleep oscillation in particular occurs very early on in the pre-symptomatic phase of neuropsychiatric and neurological disorder. This is the case, for instance, of spindle in schizophrenia, or sleep depression and sleep fragmentation, bipolar disorder, or depression. Right. So there is really an interconnection between sleep and those disorders, and there's also a spiral in the sense of if the sleep worsens during the pathology even accelerates. So we believe it's really important to understand what exactly is governing sleep to hopefully cure one or the other. And so the way we study sleep in the lab is by looking at the connectivity, dynamic, and plasticity of sleep-wake circuit. And we use different techniques, but today I will focus on single cell dynamics. So this is a classical sleep-wake cycle in human. With wakefulness, stage 1, 2, 3, 4. Now it's classified in 3 stages, non-REM sleep and then REM sleep occurring. And directly you can see that those stages can be differentiated based on EEG and EMG signals, spindle here in stage 2, and then slow wave in stage 3 and 4. And then REM sleep with a brain activity is quite similar, as one used to say, to wakefulness. So the nice thing is that you can look at the trace here, and those are the mouse EEG and EMG recording during wakefulness, non-REM sleep here, and REM sleep. And you don't need to be an expert to see the difference between that EEG here during wakefulness and And this one here during non-REM sleep, you see those slow wave and delta wave. There is even a spindle here that we can see in human. And this is the strong theta rhythm, 6 to 9 Hz during REM sleep that we can also find in human, although lower frequency. So that to say that the model that we are using can present all this EEG-MG feature that we can track, and by tracking those, we can define sleep state in a pretty similar way as we do it in humans. This is another way to represent the sleep-wake cycle. From wakefulness, they are transitioned to non-REM. And then from non-REM, they transition back to wakefulness or to REM sleep, and then exit from REM sleep goes to wakefulness. There are several non-REM sleep stages in human. There is only one so far in mice. I think it's mainly because of technology that is still not there, but I think there are efforts now that try to differentiate those states, and I think we'll succeed soon. But what I wanted to show you in that slide is a list of nuclei and very specific cell type in those nuclei whose activity correlates with wakefulness, non-REM or REM sleep. And those are the green, the blue, and the dark blue. I don't want to go into the detail, but I want to show you one thing, is that the color one are the usual suspects, so to speak, the locuses, norepinephrine cells, or the hypocretin neurons are strong wake neurons. There are some anterior hypothalamus neuron driving non-REM sleep, and some brainstem and few hypothalamic population driving REM sleep. For most of those, there is correlation, meaning that those cells are active during the specific states. But with the optogenetics or pharmacogenetic approach, now many groups have confirmed a causal link. I mean, if we activate those cells during non-REM or REM, we induce those transitions causally. That means activity in norepinephrine cells or hypocretin cells is causally involved in transition to wakefulness. I'll come back to this. But interestingly, there's a whole bunch of other cell type and nuclei that have been associated with those states, and mostly with causal approach. So this is one of the complexity of the field, is that we have a list of neuronal populations that are distributed across the brain, and we still have a difficulty to understand how they further the connectivity of those cells might be described for some of them, but not completely, and therefore we don't have the model or that really include all of those. So lots of work is needed there. Yet there is some model and currently two views that try to provide a framework to study sleep. One is so-called a circuit view, where and it's probably a more older model in which sleep-wake circuit promote sleep and wakefulness. So in that view, there are specific cells here. We talk about norepinephrine, hypocretin, histamine, that once they are active promote wakefulness, and when they are active, they inhibit the other sleep promoting one. And during non-REM sleep, vice versa, there's activity in the anterior hypothalamus, and those inhibit the wake system, right? And there's similar interaction between non-REM and REM. So there are switches, and there are mutual inhibition between those switches. There's another view that is based on the oscillatory nature of sleep, and that considers that sleep is an emergent property of cellular networks. So basically the idea here is that there's many different oscillating networks that are distributed in the brain, and sleep can start in a so-called bottom-up fashion, where oscillation can start very locally and then reach larger part of the brain and eventually generate a full blown-up EEG that is the feature of non-REM sleep, for instance. And so this is more of a local to global control of sleep. I don't have really time to talk about this today, but I'm happy to discuss it later. I think in many cases when there are different views like this, there are experiments that show that they are actually coexist. So there are, interestingly, some circuit that generates oscillation, like spindle oscillation, for instance, that once they are pushed a little bit, are able to induce transition from non-REM to REM. So really, probably those coexist. I haven't told you, but you probably know that there's a circadian regulation of sleep, right? There's also a homeostatic regulation of sleep. The sleep you lose needs to be recovered. I told you that there is a lot of different cell types that release many different transmitters, neuromodulators that are distributed across the brain. And that suggests that there might be some redundancy amongst the circuit that control either wake, non-REM, or REM. We're not sure it's, I mean, people consider there is redundancy, as I'm more on the other side, considering that there is rather a specialization in those circuits. So some circuit does something special, we just don't know what exactly yet. The oscillation view of sleep is actually based on the characterization a little more than 10 years ago that although sleep is a global state, it can be locally regulated, right? So those slow waves, for instance, here, can be locally regulated in some part of the cortex in an experience-dependent manner. And then finally, although there's an agreement that sleep rely on very distinct neuronal or cellular circuits, those are organized in a very loose network because they don't present this very nice structure of the hippocampus or cerebellum or cortex, where anatomically you can gain a lot on the functioning of the circuit. In the case of sleep-wake circuit, that's not the case, making it very difficult and probably one of the reasons why we still don't have a unified model. So I don't need to convince you that the mouse is a good model to study because the brain structure are strongly conserved, but also because we have access to different model, genetic model. I won't talk about those one, but we are using Cree driver lines, and you all know these very precious tools. The physiology of the brain, the body, the EEG, MG, EOG are very similar between mice and human, which makes this model extremely translational. So the way we organize the research project in the lab is basically around 3 points. One is really focused on the circuit that control the sleep structure. The sleep structure is sleep architecture, the transition we just talked about. And there's other project that focus on sleep oscillation, or thalamocortical generate slow waves, spindle, how they are important in sleep-wake. Rhythm, I mentioned a little bit about the work we did with Sylvain Williams and McGill and the septo-hippocampus circuit and the theta rhythm. And we do all this with the perspective of trying to understand what sleep is good for and what are the brain functions selectively or specifically associated with some of those states. And those are the questions that we have, not only us, but a lot of people in the field. Regarding the structure of sleep is what are the cellular mechanisms underlying sleep-wake states. We like to cut the behavior in initiation, maintenance, termination. It helps us with not only with timestamps, but also to dissect the circuit involved. What is the temporal and spatial organization of sleep? So that means how the brain actually does sleep. We focus on single cell, and some of the labs are trying to really bridge from single to whole brain scale imaging. And I think this is quite interesting. We discover a lot of new aspects of sleep. And in this case, I think the imaging technology are quite interesting, and applying to C. elegans or flies or zebrafish, for instance, they might provide a new way of looking at sleep in those organisms where obviously. And then eventually want to gain more knowledge about what is the sleep structure function relationship, what are the sleep rhythm good for, what is the plasticity associated with sleep or wakefulness, and how this is associated with memory consolidation. I shouldn't put consolidation, I should talk about more plasticity. It's not only consolidation, but I'll get back to this. So the tools that we are developing are both correlational and interventional, in the sense that, speaking, we want to know how the brain plays the music before we use the OCUs to change its activity. So we use EEG, LFP, or single cell imaging either with 1 photon or 2 photon imaging, high density electrophysiology, and then we can access the single cell dynamic. And then once we have this, we use tools such as optogenetics to control the activity of those cells, change the activity of some circuits, sleep oscillations, and then gain insight into some of the functions. So 10 years summarized in one slide is really focused in trying to understand the role of the lateral hypothalamus on the sleep-wake control. I go very quick on this, if it's not the main purpose of the talk today, but the first work was focused on driving CHR2 expression in hypocritic neurons, where we show that if we activate them, we basically increase the probability of wakefulness by 50% when it's unilateral, bilateral goes down to 5%. So putting really hypocritin as a neurons are major switch between non-REM and wake and REM and wake. So in the hypothalamus, these other neurons are called the MCH, melanin concentrating hormones, not a peptide produced by some cells, but they are different from hypocritin cells. And those were supposed to promote sleep, in particular REM sleep, which we so observe and confirm with optogenetic. If we drive the activity of those cells, we really push that transition here towards REM sleep. And then very naively, since those ones are also releasing GABA, we thought that GABAergic tone from the lateral hypothalamus would be a major or predominant system to induce REM sleep. We thought that activating GABA cell in the whole lateral hypothalamus would drive the animal to REM, but it just the opposite. It drives the animal to wake, non-REM to wake, but it has nothing to do on the transition from REM to wake. And this is because those cells, the majority of those cells, project to the reticular thalamus of the RTN directly, where they inhibit and change the dynamic indirectly of the thalamocortical and induce rapid sleep to wake transition. So interestingly here, those are restricted to non-REM to wake transition, not REM to wake transition, suggesting that there is specialization here again, rather than redundancy. And so we got interesting in the REM sleep or paradoxical sleep, and that's the main focus of my talk today. And there are several. We like to dissect REM sleep by its features. So there is theta rhythm in the hippocampus. I get back to the circuits that are located both in the lateral hypothalamus. We just talk about the MCH neurons, but also cholinergic cells and GABA and glutamate cells in the brainstem that are very important to switch the brain to those states. And all those brain activities somewhat associated with dreaming and imagery mentation during sleep in REM sleep. In human, how this happens is still not clear. Another interesting observation is that there is a tonic muscle atonia during REM sleep, and this has been described by Jouvet in Lyon and Pierre Vellupi, where there's inhibitory of the tonic inhibition of the motor output during REM sleep that really block those movement, and if this inhibition is released, the animals enact their dream. It's quite interesting. Interesting in these days, a lot of video on YouTube. And then one aspect that Carolina in the lab next door and then us were interested in, together with Marco Celio in Fribourg in Switzerland, is what generates those eye movement during sleep, rapid eye movement. The function is still not known, but we found some nucleus in the brainstem that seems really to be important in generating those movement. But again, to show that we can try to dissect all the brain mechanisms behind each of those REM sleep features. And one that we did in collaboration with Sylvain, and that was the work of Richard, was to try to understand where the theta reading is coming from during REM sleep. So this is a schematic of the circuit driving theta rhythm in general during wake and REM sleep. And the question really emphasized three different populations here: glutamate, GABA, and cholinergic cell in the medial septum projecting to this hippocampus, being involved in changing that rhythm. And to make a long story short, we found that the GABA cell in the medial septum are responsible of driving those theta rhythm if we silence them here. the theta rhythm in the dorsal C1 is completely gone. This is how we drive the experiment here with, I think this is 10 seconds of orange line. When we silence the MS GABA cell, the amplitude of the theta is strongly decreased. Interestingly, only the theta rhythm was affected. None of the other oscillation, including the gamma and the ripples, were changed, not the EMG tone. So we thought that would really provide us a nice way to modulate brain activity during REM sleep and see what this brain activity is good for. And that's what we did, not with a novel object, or we did it with a novel object recognition test, but I'm showing you the fear conditioning here, where animals were trained on a fear conditioning and then put back into their home cage. And once they were in their home cage, every time the animal entered in REM sleep, we would turn the laser on, silence the GABA cell, and when the REM sleep stopped, we turn the laser off. We did that for almost 3 to 4 hours, and then let the animal sleep, let them be awake during the night, and then on day 2, we retest. And this is the retest of the animal in the same context, but no electric foot shock, no auditory cue. And you can see that the YFP control, as expected, freeze, but the animals in which MS GABA cell has been silenced don't, suggesting that they don't recognize the context. This is quantified here. The green one here are significantly different in the freezing duration, suggesting that they didn't consolidate the contextual memory here during REM sleep. They are still able to discriminate and react to the tone in a very similar way than the control animal, as you can see here. And this really goes with all the literature, because the context is included in the hippocampus, which we are indirectly manipulating here in that experiment, while the discrimination of the cue here is encoded in the prefrontal cortex. So that was the main message of that first study, where besides the mechanistic finding that we made, we found that the activity of those medial septum GABA cells during REM sleep are key for consolidating contextual memory. So that raised a lot of different questions, whether there's subpopulation of GABA neurons in the septum, what does the non-REM, which also encode context through place cells, for instance, do that the REM doesn't, or also do? We don't know. This is still an open question in terms of contextual memory consolidation. But one aspect that we were quite interested in is what about the emotion and the discrimination of the auditory cue? And that's something we pursue in the next study that I want to show you, and we published recently. And again, we start that study not with that question primarily, but with another question of which is the following one. What was the mechanism underlying cortical activity during REM sleep in the prefrontal cortex? So, and then what is the implication? And we start from this type of trace, right? Where the EEG is showing this massive and very stable theta oscillation. So we wanted to understand what, obviously this comes from the hippocampus, but what does it override in the cortex? And this work was done by Mattia and Nicolo for the data analysis. Thiago helped us with the patch. And I'll show you some unpublished data from Michaela, who's taking the lead now on that project. So I think I like this meta-analysis here, because it's in human, but it collects 10 years of work using imaging technology in human, and the blue dots and square show the brain structure, brain area, nuclei whose activity is decreased during REM sleep, while the yellow one is increased. So basically, prefrontal cortex is massively decreased, while limbic system is strongly active. So that shows you the diversity of activity, and also the fact that in the cortex there's many different heterogeneity in the response. And this is shown in an old study from Timofeev, Igor, what they called single unit of pyramidal neurons or interneurons, again during REM sleep, and could differentiate different class of neurons without being able to know exactly what those neurons were, but at least suggesting that they are still active and silent cells in some layer of the cortex. And more recently, the colostrum here has been shown to be extremely C-positive and active during REM sleep. This is ongoing activity. I think Romain is working on it. So the preparation that Mattia developed was the head restraint two-photon imaging. So we trained mice to sleep with the head restraint under the two-photon microscope. So that takes between 10 and 15 days. And those animals were injected with a virus that target GCAM6 to pyramidal cells using the campkinase 2. Those are the cells that you can see here. Once the animals were trained after 2 weeks, we could image the activity of those cells in layer 2, 3 across non-REM, REM, or wake, called the sleep-wake cycle. Here you see the hypnogram here with wake, non-REM, and then some REM. And interestingly, we found that during REM sleep, there is a massive decrease of the activity of those pyramidal cells. So this is a heat map of each of the cells recorded here. Those are the delta over F single traces, and it's quite consistent across animal REM sleep. So that means that all the REM sleep, all the animals that we have recorded show very similar results. So there's a decrease in the cell body of the pyramidal cells. The next question was, okay, what is the activity of the interneurons in the cortex? And so, you know, there are PV interneurons, VIP interneurons, and somatostatin interneurons, and they are organized in a very particular circuit. In the way that PV cells provide inhibition to the cell soma, while the VIP usually inhibit somatostatin and inhibit the dendrites. Right, so there is this circuit. You'll see that it's not always that canonical sequence, but we based our work on this, and we imaged the PV cell and found that selectively during REM sleep, those PV cells are massively active. They really provide a strong inhibition to the circuit. And that was also the case for the VIP interneurons, right? You can see here. Oh, of course, not all the cells are active. You see this pretty much 80% active. Some are not. So there is some variability, but there's a strong inhibition of those VIP cells. And if we follow that circuit, we also observe that the somatostatin are quite silenced, and that might be due to the excitation of to the inhibition of the somatostatin cell by the VIP cell. I lost it. So that's the stats per animals. There's a decrease in the pyramidal cell body, increase of PV cell activity, increase of VIP cell, and decrease of somatostatin. So all this, based on this circuit here, suggests that if the cell body are strongly inhibited, the dendrite might be quite active. So that's what we did in the next step. We imaged the dendrites in layer 1, so a little very superficial layer doing REM and non-REM. And as you can see here, we found some dendritic events during REM sleep here that are quite localized. We found some that are quite larger here. And of course, they are not restricted to REM sleep. We found some during wakefulness and also during non-REM. But interestingly, we found that both for the spreading event and the local event, they were quite high in REM compared to non-REM and even compared to wakefulness, right? Suggesting that there is a huge dendritic activation during REM sleep, while the cell body is maintained strongly inhibited. Where does that inhibition come from? I told you, well, this is VIP cell inhibiting somatostatin cells. The reviewer asked us to show this. So what we did in that experiment was to use switch R to silence VIP cells and see whether that would release somatostatin cell activity and therefore dendrite inhibition. And that's actually what we observe. When we turn the light on during REM, you see that the activity of the dendrite is almost down to 0. Probably because the somatostatin are exerting their inhibition again on the dendrites, and this stops when we turn the switch R off. So basically demonstrating that the VIP cells are really key into this somatodendritic decoupling. So what could drive those change in the excitation inhibition during REM sleep? So one way that we took to study that was to map the input to those cells using those Cree-dependent virus. I can talk more about this later if you want, but basically you use a combination of two viruses, one that label the cells in green, that will be the starter cells, so the input cells, and then the red will be the cells that project to those cells. It's the opposite. The starter is red and the input are green. And this is shown here. Those are the input, and those are the cells that project to this prefrontal cortex. So it's not surprisingly retrospinal cortex, somatosensory cortex here, but interestingly, the CM, the central medial thalamus here. Quantify here for both input to PV cells here in red, VIP in green, or pyramidal in black. And we found that the PV cell receive a massive input from the thalamus during REM sleep, also from other brain area. But we focus on the thalamus because we know from another study that those CMT cells, they are strongly active during REM sleep, even more than during wake and non-REM. So the hypothesis here is that probably they might be the one driving the PV cells. So that's what we wanted to see. So anatomically, we have monosynaptic projection from the CM to the PV cells. This is a brain slices experiment showing that if we drive CM cells with CHR2, record the PV cell, we found the inward current, suggesting there is inhibition with short latency, suggesting not in all the cells, right? Not that there's half of them maybe that are responsive. That's not surprising, but that might be sufficient to drive the activity of PV cells, right, during REM sleep. And so the experiment, before that, we wanted to show that the axon of those thalamic cells in the cortex were also active during REM. And so this is what you can see here. We target GCAM6 in the cell body of those cells, and then image the axon of those cells in the layer 2-3. And interestingly, we found that they were strongly active during REM sleep. So really all the elements are there to suggest that those are the input driving the PV activity and therefore the inhibition of the cell soma. And then we show that there was revealed two questions. We ask us to silence the thalamus here while we image the pyramidal and the PV cell. So we image the pyramidal in green and the PV cell in red in a quite tricky experiment. And during REM sleep, if we silence the thalamus starting here on the blue line, you can see that there is a decrease of the activity of the PV cells here and an increase of activity of the pyramidal cells here. So those are the heat map. This is the delta F over F. You have to compare the reactivation of the pyramidal cell body here to the one that I showed you at the beginning of my talk, right, where there's very few activity normally. So really, this suggesting that is one of the main input and driving force of the PV inhibition onto the pyramidal, and that this comes from the mediodorsal thalamus. And so in summary, this was, I think, quite a surprising discovery to us. The fact that there is this somatodendritic decoupling during REM sleep in this part of the cortex, prefrontal cortex, almost boundary with motor cortex. I'll show you that the mechanism here involves local interneurons, right? That by inhibiting PV inhibition really block the output of the pyramidal cells. They are not able to fire much during REM sleep, and the VIP to an inhibition of somatostatin provide a disinhibition of the dendrites with both local and global propagation across the dendritic arbors. And that provide probably a lot of integration during REM sleep. As a sleep researcher, it was quite interesting to see the parallel with how REM sleep and more paradoxical sleep is usually described in the field as states where the brain is wide awake, but the body is completely shut down. And that's it reflects, I mean, that's due to the strong brain activity and the muscle atonia. But we basically have the same here at the single cell, right? The cell body is completely shut down when the dendrite is wide awake. So what that can provide to the circuit in terms of potential plasticity or behavior, or what is this good for? And so there is many different functions associated with REM sleep, also with non-REM sleep, but today I focus on REM sleep. I listed some of them here. Energy allocation is one of them. Development on sensory motor, and babies do a lot of REM sleep, and all this muscle twitch and sensory input are thought to really contribute to the development of the somatosensory cortex. They are involved in learning and memory. I showed you one study, as many others involved in that. And in emotional regulation, and that's something we wanted to focus on with this particular circuit, and for good reason. I'll show you this. And on the right, I put really our view on what sleep can do in general. Before we go at the synaptic level, I think at the behavioral level, sleep might be a really important state for daily update that will prepare the organism to future challenges, and really provide a synaptic to behavior optimization. And this is important for any organism, but on a long time scale, that might be important for the survival of the species. In other words, that means that you can sleep. You can sleep without REM sleep. Whether you will be fit to survive, that might be another question. So how we test the behavioral implication of these somatodendritic decouplings by using the fear conditioning. It's a little different than the one we used before, because in this one we had CS plus, CS minus. That means there's a sound cue, the CS plus, that is associated with a food shock, right? Well, the CS minus is not. And so there's really a danger signal versus a safety signal. And so we run those in two groups of animals, one in which we silence PV cell and one in which we silence VIP cells. And we focus on this because by silencing PV cell, we let the pyramidal cell fire, where they're normally shut down. And by silencing VIP, we restore the somatostatin inhibition of the dendrites, right? So we just shut down the dendrites. So we basically reverse it during REM sleep. And again, training on conditioning here, and then right after conditioning, animal is back to its home cage, and we silence selectively during REM sleep. No change on sleep-wake cycle. I should also have mentioned that for the other study. But interesting, look at what happened when we silence the VIP cells. So we have the control in green and the RHT VIP, which is the one we silence the VIP cells. The green one freeze to the CS plus, not to the CS minus, right? This is what is expected. They learn very nicely that the CS plus is associated with a food shock. But if we silence VIP cells, I mean, if we shut down dendrites, right, the animal is unable to discriminate. So they hear the sound, and they freeze similarly to both the CS plus and the CS minus. So there is no, the discrimination is lost here, so to speak. But in contrast, when we silence PV cells, again, same control RHT animals, the control learns, and then the RHT, I would say, learn even better, right? So they discriminate the CS plus and CS minus, but they freeze even more to the CS plus. So if you look at discrimination index, they really goes in opposite direction, right? The VIP cells don't discriminate CS plus, CS minus, the PV cell over discriminate those and over freeze. This is actually associated with synaptic and opposite synaptic plasticity in the pyramidal cell there. So this is an experiment that was done with by Tiago in the lab. And the experiment is basically shown here. I didn't mention it, but after the conditioning, we patch the animal right after the opto-stim during sleep. And so the brain slices showed that if we silence VIP, there's a decrease of AMPA and MDA ratio, suggesting a downscaling when there is no dendritic activity, while an inhibition of the PV increased this AMPA and MDA ratio, suggesting an increase in synaptic upscaling potentially at the single cell level, right? And this is actually nicely correlates, so this AMPA and MDA ratio for the VIP and the PV nicely correlate with the discrimination score of the animal. So really showing that this somatodendritic decoupling is important for opposite synaptic plasticity in the cortex. So in summary here, Besides the somatodendritic decoupling, I think we found that they regulate discrimination of those cues in this case in really opposite way, right? And the VIP is really important for discrimination of those cues and the absence of those VIP. Disinhibition of the dendrite is probably responsible for the generalization of the behavior. Other circuits might be involved, but that suggests that basically during REM sleep and in association with the dendritic activity, there is a strong synaptic upscaling possibly, while there is a downscaling during due to the shutdown, the PV mediating shutdown of the pyramidal cell bodies. And the cell body are unable to exert the usual top-down control on subcortical brain nuclei during REM sleep. So this, of course, relates to many neuropsychiatric disorders, including PTSD, where the clear over-consolidation to CS+, but also panic and anxiety disorder. Whether this is the mechanism, we don't know, but this is at least one effect. So we still have some questions. I don't have data on this per se, but we would like now to link the contextual information with the cue discrimination from the hippocampus and the prefrontal cortex, respectively. So, and one brain area that might help in doing this is the retrospinal cortex. Why? Because anatomically they are connected, but they also show this strong activity in human and in mouse during REM sleep. You see here the retrospinal cortex is massively active during REM sleep. This has been shown with CFOS approach, so that's quite a good candidate. This is ongoing work, but one question that we quite quickly had in mind is whether the somatodendritic decoupling is a general mechanism across all the cortex. So I haven't shown you the data on the somatosensory cortex, because we have few animals that show exactly the same mechanism, and this is the primary data from the retrospinal cortex, where we also see a decrease in the activity of the pyramidal cells here significantly during REM sleep, and this goes with an increase of the dendrite activity of those cells again during REM sleep. So we believe it's quite conserved, this phenomenon. The PV, somatostatin, and VP and VIP cells are quite similar in the sense that the PV cells activity are also high during REM sleep. The VIP cells are also high, but interestingly, the somatostatin are quite high, and this is quite interesting because it's an opposite result as what we have in the prefrontal cortex. Where does that come from? We are not sure. Probably a circuit might be differently organized. One aspect also that we are looking at is some delay in this change of activity. In the previous study I showed you in the PFC, all the change really occurred at the onset of REM sleep. But here, in the prefrontal cortex, there is up to 30-40 seconds during which the pyramidal cells are still active, or the somatostatin are still not really active. And so there is this delay that is consistent with across cell, across REM, and across animals, that suggests that there might be two different states within REM sleep. What they mean, what it is, we don't know. Whether this is linked with acetylcholine modulation is something we are looking at. It's not the case for PV and VIP, so maybe there is compartmentalization in the dynamic of the circuit. But something that is quite encouraging is to see that this somatodendritic decoupling is actually also present there. So now the next question is whether this also happens in the hippocampus, because similar circuits are also because the circuit in the hippocampus and their activity during REM is quite similar. There is probably some different local connectivity in the sense that VIP are not inhibiting somatostatin in the retrospinal cortex, and that might be due to local connectivity or cholinergic modulation or input from other subcortical area. Whether this delay is important for information transfer, for instance, is something we don't know, but that's what we would like to test. Last slide is this such a surprise, right, to find a somatodendritic decoupling? Yes, Yes and no. Yes, because I think it's never been shown in REM sleep, but no, because it's been described for layer 5 here in this paper from the Larcum, Mathieu Larcum labs here during anesthesia, where during anesthesia there is this somatodendritic decoupling, and that he has a model where he suggests that this might be responsible for the lack of consciousness anesthesia, because there is no connection between feed-forward and feedback inhibition. Other group in Lyon have shown quite similar profile of cell activity during REM sleep. And finally, there are some old model that suggest that the link between REM sleep and dreaming might be a model of proto-consciousness. So the idea behind this is that REM sleep is a state in which there is not a complete loss of consciousness. You know, if you remember your dream, if some people are able to do lucid dreaming or take decision during their dream. So there is a level of consciousness that in some studies have been associated with strong activity in frontal part of the brain. We see here in REM sleep or in lucid dreaming. So whether this type of huge dendritic activity and in the cortex, in the prefrontal cortex, contribute to this, we don't know. I think talking about consciousness in mice is difficult, but it's interesting to speculate. And I'll finish my talk here by thanking people in the lab, Mattia, who did the work, Thiago, Nicolo, and Michaela, my collaborator, and the funding agency, and you for your attention. Thank you.